a God of the gaps argument. Too complex to explain by natural processes, therefore must be a designer. And as Michael said it so nicely, and I agree, I can't figure out how to explain it, is what they are basically saying, therefore no one can, which is pretty arrogant, because eventually somebody will. Okay? This is not a legitimate scientific way to tackle a problem. There are many, many unsolved mysteries in science. That's a good thing about science. It's always open-ended, okay? We don't just give up and say, oh, can't explain it by science so far, therefore we should fall back on supernaturalism. That's a cop-out, okay? That's a non-explanation. Now, the problem of the origin of life is admittedly a very difficult problem, and scientists have been working hard at it for about 50 years now in intense ways. The major problem, of course, being that there's the original evidence of it. It's all chemical, which is not something that fossilizes very well. But despite what you might hear from creationists who ridicule and trivialize it, enormous progress has been made on the chemical side of this. And as I'll show you, enormous progress has also been made on the fossil record side. We don't yet have life in a test tube, but there are several labs that are very close to it as I speak. But just because we don't have all the pieces of the puzzle yet doesn't mean we give up and say, too complicated, therefore I don't believe it's going to be proven. The experiments run all the way back to the famous Miller-Urey experiment in 1953, which showed amino acids are very easy to build. In 1 week, he made 4 of the amino acids of life. His original gunk was found in his freezer after he died, and now there are 22 amino acids found in it. Later experiments have produced nearly all the 20 amino acids found in life, and now they've looked at meteorites, and it turns out amino acids are everywhere in the solar system. They're incredibly easy to make. The next step in the argument, of course, is to link amino acids together to form proteins. This has been done by many different research labs over the last 50 years, going way back to the 1950s. You can splash amino acids on dry rocks and polymerize them to proteins. You can react them with cyanide, with clays, with heat. Proteins formed in lots of ways. The cell wall that surrounds every simple cell is a series of bipolar lipids, which are naturally polarized. They have this water-hating end right here in the little tails you see, and a water-loving alcohol on the other side. They naturally line up just like when you mix Italian dressing, right? Oil and water droplets, and form little droplets which have the same properties in many ways as cells. Nucleic acids are by far the most complicated thing to develop, but they've been synthesized in lots of ways, by heating in cyanide, by UV radiation. A paper just published a few months ago by Sutherland et al. Actually created ribonucleotides and very simple early Earth chemicals, and they were fully complicated and fully operational. Many, many experiments done on how you do these things, wetting lipids to produce droplets, to then trap DNA and concentrate it 100 times. Famous experiments by Potton and Sidney Fox that made proteinoids which bud, change size, absorb and release compounds like bacterial feeding, excrete waste products, even metabolize starch. The only thing those things don't do is reproduce, and with DNA. Here's some images from Sidney Fox and from Potton. And the beat just goes on and on. If you just follow the literature and read any of the stuff in the science press, stuff has been accumulating on almost a weekly basis for at least the last 30 years. The coding sequence of amino acids is easily built with small RNA templates in normal prebiotic conditions. The first ribosomes in the RNA world were much longer and more stable. Cyclic nucleotides can easily emerge in water to form RNA over 100 nucleotides long. RNA molecules link up in long chains easily under normal Earth conditions. Several people have shown that the Urey-Miller experiment was right in the way that they modeled atmosphere, and even if it weren't, it still works, even in different conditions. And you'll hear these people say, oh, there are no new genes being produced. This has been done repeatedly in labs all over the world. Now, I'm a paleontologist by training. I'm one of those kids who got hooked on dinosaurs at age 4 and never grew up. Okay? And all my life has been focused on fossils, although I had a lot of background in biochemistry as well. And there, of course, the evidence is ironclad. Huge amounts of evidence, none of which these guys will ever treat very fairly. We have fossil record going all the way back to 3.5 billion years of the simplest bacteria. And then, as you would expect, evolution predicts from simple to complex, and sure enough, the fossil record shows that in spades. 1.7 billion years ago, we have clear eukaryotic cells, which have a nucleus. 600 million years ago, we have the evidence, the earliest phyla, the sponges, the corals, the arthropods, and so on, way before the Cambrian. At 580 million years ago, we have soft-bodied multicellular organisms with no skeletons yet. In the earliest Cambrian, 550 million years ago, we get these so-called little shellies, which have very small pieces of skeletons, and not until 520 do you get things as big as a trilobite. I'll show you a few images of these just to do this quickly here. Here's some images Bill Schopp at UCLA loaned me. These are various bacteria and cyanobacteria he discovered in Australia. And over here on the right is the modern cyanobacterium Lyngbia, which is a dead ringer for these fossils. You'll hear our opponents probably say something along these lines. This is from Meyer's 2003 paper. The body plans, the structures present Cambrian period animals have no clear morphologic antecedents earlier strata, and the Cambrian explosion lasted only 5 million years. And this is a figure from their paper. It's false. Okay, and I'll show you why in a second here. I'll say this statement is also false. It's not a Cambrian explosion. That's an unfortunate old-fashioned term that's completely out of date. I like to use something a little more modern, like Cambrian slow fuse, which is really what it is. I'll show you this evidence very quickly here. We have it 600 million years ago, the first evidence of multicellular animals in the forms of embryos of sponges, cnidarians, worms, and arthropods. Almost all the major phylum marine invertebrates that have a fossil record 50 million years before the Cambrian. Okay? And at 580 million years ago, shortly thereafter, we get these big multicellular soft-bodied things, the so-called Ediacarofauna, which are worldwide and known from many different kinds of interesting localities. They're soft-bodied, but they're quite large, but they don't have skeletons yet. In the very beginning of the Cambrian, before trilobites, we get these little shellies. These are mostly a millimeter or two across, and you can see the ones up here are very clearly mollusks. Many of these are spicules of sponges. They're all members of phyla, which we have recognized from later in the Cambrian, and they're all well before the complex so-called Cambrian explosion. If you actually go to the outcrops, which I've done myself, and I doubt that either of my opponents has ever seen or collected, like I have, this is the outcrop in Aldana River in Russia. You can see this entire sequence right in the cliffs as you climb up from the bottom to the top. It's a very long, protracted event, taking over 80 million years. So Cambrian explosion is not 5 million years long. It's from here all the way down to here, which is well dated to 80 million years. And that's hardly explosion by my standards. You'll hear my opponents talk about information. Okay, they constantly say life is too complex. Something had to insert information into it, put it in, make it of some kind of designer. And once again, that's a God of the gaps argument. Okay? Moreover, there are many biological and non-biological systems that assemble naturally from simple to complex. Here's a classic example, a very simple reaction which some of my coaches suggest I actually perform on stage for you. It's known as the Belousov-Zhabotinsky reaction. All you need is an organic solution with some bromates, some cerium sulfates, citric and sulfuric acids, add a catalyst, and boom. You get a reaction that's self-propagating and has a cyclic nature, just like the Krebs cycle, and keeps going indefinitely. And several of these ID creations, like Michael B., has said the Krebs cycle cannot be explained by normal chemistry. This is exactly like it. Or you'll hear my colleagues say, or my opponents here say, information. So let's pose a question. What do we mean by information? What kind of systems are we talking about? They're going to talk about biological systems. Well, let's look at this question. What kind of natural system builds up from simple to complex, has mutations, replicates those mutations, gets much more complex, and increases its information? And you probably think you know the answer. No, you don't. The answer is mud. Turns out, if you go look at common mud, it's made of simple silicates known as clay minerals, which combine very easily by electrostatic interaction from very simple little tetrahedra to chains, to single double chains, to these big sheets. And then once you get them into sheet structures, you get very complicated structures that go indefinitely for very long distances, up and down the screen and right to left and in and out of the screen. These are all sheet silicates. You can see on the lower left there what they look like. They look like little books on side view. And they get bigger and bigger and propagate themselves. And more impressively, this has been shown by a number of clay mineralogists, including friends of mine, when they have lattice defects or weird substitutions or so on, those are reproducible. The clay minerals that grow from this template will reproduce the same mutation, the same defect, and grow with that mutation and carry on. So you can have these things growing on, and they'll keep on growing as long as they have the right conditions, until you get very large self-reproducing systems with mutations and replication. The only thing they're not is living. So we hear them say over and over again, complex biomolecules require design. Well, actually, clay minerals are more complex. They mutate and they replicate. So if they're going to apply that standard, then we're going to have the intelligent designer create every drop of mud individually. So I will wrap up here. I don't know how my time is, but I'll wrap up my time here. As Michael said, we have some issues here, but one of the big issues is, are these guys playing by the rules of science? Have they done any serious science? And I think we'll show tonight they did not. Virtually all their scientific research is borrowed or copied from other people. I don't seem to see any example of real lab work by these people. Their basic publications are criticizing works of real scientists. I never see any of these guys at scientific meetings, and I go to all of them each year, okay? A very limited publication in peer-reviewed literature, and I'll talk about that later if it comes up in questions. And more revealing, as Michael pointed out at the beginning, what's really behind this is nothing about science at all. All these people have, one way or other, revealed that their whole dogma is driven by religious motives, which is not a bad thing in and of itself, but it has nothing to do with science. And before they took it off the web, but you can find it very easily, it's all sorts of places to find it on the web now, they have what they call the wedge strategy. It was a very clear document issued by Meyer's own institution